We just started.
Good morning, everyone. It's really nice to have you here again for our third webinar of the Greek Scientist Society. Today we have the honor and the pleasure to ones that do not know the exact whereabouts of the professor. I would like to introduce him shortly, saying that he's currently an MIT professor, having spent previously some years at Princeton University, and his main research areas are biophysics, computational modeling, microbial systems, synthetic biology, and tissue engineering, all combined into an effort to apply them for innovative gene regulation systems, gene therapy, drug development, biocomputing, and even bioremediation. So I would like to welcome you. First of all, I would like to ask Theo Zacharis to welcome everybody. Theo?
I may face some problems. I don't know. I hear my voice back.
Okay, then maybe it would be better to give the stand to Professor Weiss. Yes, Professor Weiss, thank you very much for being here today. Please take the stand. We're all looking forward to this presentation.
Great. Thank you for inviting me. I'm going to share my screen now.
Yes.
I think you should be able to see it, and I'm going to go to full screen mode.
Yes, so, it works fine. Thank you.
Okay, excellent. So thank you. It's a pleasure to be here. Thank you for the invitation. And I look forward to discussing with you mammalian synthetic biology. So I'm going to focus on foundational aspects of mammalian synthetic biology, as well as therapeutic applications. So I'm actually a computer scientist by training. And about a quarter century ago, as a graduate student at MIT, I became fascinated with this notion that we might be able to program cells in the same way that we program computers. And so when I think about a cell, what I get excited about is this notion that I can write a biological program. So it looks like a computer program, but it's meant to actually program a cell. And what it does is it allows me to express specific behaviors that I want the cells to be able to perform. And it does so in a formal way, without being vague about it. And so I can take this description of a program or behavior and then convert that to a regulatory network. This would be an abstract regulatory network that would be something that in principle might be able to be implemented inside the cell. And when this network is put inside the cell, the point is that it allows us to reprogram what the cell is doing. Then I take this abstract regulatory network and I convert it into an actual piece of DNA, a physical piece of DNA, which then I can put inside the cell. And by doing that, I've just been able to reprogram the cell with new behaviors. So this notion of reprogramming the cell really is essentially my definition of what we'd like to do with synthetic biology. Now, when we think about reprogramming cells, it's important to understand or try to understand what it is, what is the substrate, what are the properties of the substrate that we are trying to reprogram, and really appreciate the complexity of the biological substrate. So here I'm showing you 3 examples of very complex networks, and I'm obviously not going to go through the details of them. It would take possibly years, but the point is that these are networks in cancer, these are networks in development of organs, these are networks in liver metabolism. And so these are very complex networks, and when you begin to think about wanting to do cancer therapies, or you're wanting to control organ differentiation and generation, or wanting to control metabolism in liver, it's really important that we respect the complexity of the biological system and try to use tools that are able to match the complexity that this substrate has. And so the point is that when we can begin to understand this complexity, and we don't have to have a full understanding. It's okay, to have some partial understanding of what different factors do. That then allows us to begin to say, okay, we need tools that are up to the task. Okay, and so when you think about problem-solving tools, one thing that's a simple tool that's very common in many fields is this notion of saying, okay, let's use the 5 W's and 2 H's. So when we start a project, basically define how we're going to address this problem by asking the questions who, what, when, where, why, how much. And so imagine, for example, we're trying to do this with cancer. We might ask the question who, so this would be cancer patients, or we would want to say only tumor cells within the cancer. What? So what kinds of therapeutic proteins might we want to produce? When would we do this? So it's important to time the production of these therapeutic proteins. Where are we going to do this? So let's focus only on tumor cells and not do this for non-tumor cells. Why are we going to do this? Obviously, high level, we want to cure patients, but also we can ask it on a technical level, which is under what conditions do we want to produce that therapeutic protein? How are we going to produce it? And also, it's important to understand how much of the therapeutic protein. So these questions are really fundamental. It's important that we have some initial ideas about what we want to do prior to engaging in these particular projects. And so it's obviously an important set of questions. The question is, how do we actually use synthetic biology to do that? And what's emerged in the field of synthetic biology, we're calling it essentially the central dogma in synthetic biology, is that almost every project now in synthetic biology has these 3 main components. It has a sensing component, which is, mechanisms whereby we can engineer genetic circuits to sense what's going on outside the cell, molecules outside the cell, or molecules inside the cell. That information, these sensors can then feed into a logic processing element. So these would be genetic circuits inside the cell that can take the information, can take these biomarker sensory information, and then be able to make decisions about what to do based on integration of multiple pieces of information. And then once a decision is done, then the last component is a regulated action or a function. So for example, modify the production of genes inside the cell, or activity of proteins, or secrete something outside the cell. And then these things can also be used in a feedback regulation way, so that what we secrete can then be used to sense, what we engineer the cells to secrete can be used to sense what to do next. And so we can take these kind of high-level abstractions, or these genetic circuits that represent behavior programs, and then think about what kind of programs would be interesting to be able to implement. So I'm going to talk about many of these. We want to induce expression of genes at the right time, at the right place. We want to be able to use multi-input logic to control when we activate certain things. We want to be able to create these on-off toggle switches, so we can basically turn on expression or turn off expression where we like, and feedback regulation and so on. So these are some activities, some types of behaviors that we want to be able to engineer the cells to do. And then what we can think about is then in translating those into these gene regulatory networks. Now, as I mentioned before, the biological substrate cells are really complex, so it's important to understand that these programs that we put inside the cell are really just small little programs within a very complex environment. And really appreciate that and trying to understand how can we make reliable programs, despite the fact that we don't necessarily have a full and complete understanding of the complexity of the biological system. So now, in terms of thinking about is this real, or is this still kind of science fiction? And synthetic biology is in fact real and right now. So I'll mention a few near-term applications that we are looking into that could actually be translated into industry essentially right now, and we are in the process of translating them into industry. So one of them is this notion of controlling for these monoclonal antibodies. Monoclonal antibodies are basically therapeutic drugs, okay, that you can inject. One example of monoclonal antibodies are ones that can be used, for example, to suppress the activity of SARS-CoV-2, the virus in COVID-19. And those appear to actually, when they're used really early on, those appear to be really highly effective, okay? And so one important thing about these monoclonal antibodies is that the sugar that decorates this monoclonal antibody molecule has a major effect on its efficacy, okay? But it's really hard to control the sugar decoration, and we've been able to show with a publication a couple years ago that we have very precise ways of exactly controlling the sugar decoration on these monoclonal antibodies, which should have a real effect, right? If this was used in industry right now, would have a real effect on the efficacy of these treatments. Another one that I'm actually going to spend a good amount of time on is this notion of programmable organoids for drug testing. And the idea is that we can take cells, for example, stem cells, and be able to reprogram them to create little organoids. Okay, so I'll show you an example of us being able to create little liver organoids that we can put on a microfluidic chip and then be able to perform drug testing on it and figure out, is this drug toxic? Is it safe? Is it efficacious? So rather, or in addition to testing drugs on animals before we go to humans, it seems like it would be much better to be able to test these drugs on human-derived organoids that are sitting on a microfluidic chip. And so we have interactions right now with Big Pharma about incorporating this very soon into their drug development pipeline. And then there's another example I won't go into in terms of being able to produce therapeutic viruses. So these are examples of near-term applications for synthetic biology. And again, this is the one I'm going to focus on today. Medium term, so these are applications that would take a little bit longer, several more years, but they're beginning to make their way into the clinic. So these are some things I will talk about. These are examples that I'm showing here, being able to control the expression of particular therapeutic proteins, when they get expressed and where they get expressed. One thing that's coming up in the medium term is this notion of taking immune cells, your T cells, and being able to genetically engineer them to target cancer cells. And with synthetic biology, you can make that be highly specific so that they only attack tumor cells and rarely attack cancer cells. What I'll talk about today is a project where we are genetically engineering viruses to do something similar, where these viruses enter cells, they infect cells, and then they perform a biological computation. Is this cell that just got infected a tumor cell or not? And if the answer is yes, it is a tumor cell, then the virus ends up killing the cell. And if the answer is no, it's not a tumor cell, then the virus just shuts itself off and goes away. So this is an approach that we have right now that I'll talk about in detail for cancer therapy. And in the longer term, many things can become, are exciting to think about. Okay, so more programmable organoids that can be transplanted into patients. So, for example, that have liver disease, and these are things that we're actually beginning to try in mice right now. Or something I'll talk about is encapsulating cells into beads and then putting them into your body so they can sense if there's some kind of malfunction. And if there is a malfunction, then they quickly notice that and they can correct it. If you have, your sugar levels are wrong, or you have some impending, maybe heart attack and things. These are the kinds of things that we're thinking about. So a quick sense and response. So the way we're going to address these is going back to the 5 W's and 2 H's. We're going to think about what it is that we want to engineer, what behavior we want to engineer, by being able to create mechanisms, biological circuits, that allow us to control these things. So for example, on the where, I'm going to demonstrate how we can control expression, gene expression in specific cell types for organoids, or just in cancer cells. What are we going to express? So these are things I'm going to show that can control how stem cells differentiate, or things that can control whether cancer cells are destroyed or not, or create inflammation to help with cancer treatments. How much? So this is, even though I come from a computer science background, it's important to understand what makes sense in biology. So sometimes we want to turn things just on and off, but sometimes we want to have very graded responses. So I'll show you that. And sometimes we want to really jack up the expression levels as much as possible. So we want to be able to create amplification mechanism. And then the other thing I'll focus on is the timing. Do we need to respond in hours, or do we need mechanisms that allow us to respond in seconds? So I'll give you examples of both. So let's start with cell lineage specific expression of sulfate regulators in a graded way where it's okay to respond in hours. Okay, we don't need faster responses. This is just fine. And so this is going to be in the context of this notion of programmable organoids. And the idea is we're going to start with stem cells, and then we're going to genetically program them to proliferate and then create patterns. And then these patterns of differentiated stem cells could end up allowing us to create these organoids on a chip, or ultimately, can we create an engineered liver? Can we create mechanisms that allow us to regenerate, for example, spinal cords? Okay, so on the liver front, I'll show you data where we are now able to make some fairly interesting liver organoids. And we're going to do that with genetic programs that control various properties of stem cell differentiation. Okay, and we can do this for creating the tissues. We can also create sensors in there that can tell us, if you apply specific drug candidate, what's the response? Is this toxic? Is this affecting the right metabolic pathways, for example? And then ultimately what we want to be able to do is even genetically engineer these organoids, like these livers, to be more robust so that they can engraft better in a person. And maybe, we can even make liver that is able to resist certain virus infections. Okay, these are the kinds of things that we can already begin to imagine. But let's take basic steps on understanding how to get there. And so in terms of the organoids, what we did is about 15 years ago, we were able to genetically engineer bacterial cells to communicate with one another in a petri dish, create all of these genetically programmed patterns. And we said, okay, we now can genetically engineer bacteria to create patterns. Let's think about moving this to mammalian stem cells and human stem cells, and can we create tissues by design? And so one of the things that we set out to do was this notion of let's take stem cells, which is this ICM, the inner cell mass, and then create a multi-step biological program that would take these stem cells and one step at a time in an automated fashion, differentiate them to what happens in the way that happens and inspired by what happens in normal biological development, from the stem cells to endoderm, to pancreatic progenitors, to endocrine, and ultimately to insulin-producing pancreatic beta cells. Okay, and the point is that we want to be able to do this to be able to generate these beta cells so that we can ultimately embed them in somebody with diabetes. Right, so this would allow us to potentially address, and who knows, possibly even cure diabetes by creating patient-specific insulin-producing pancreatic beta cells. Obviously, as you can imagine, this is a fairly ambitious endeavor, and we are not quite there yet, but we have made some really good progress along the way, and I will share with you this progress. Now, the way to achieve this is, as a computer scientist, I think about this as a biological program that has several different timed steps, and basically they're these if-then statements, or if-then rules or actions. So the first action is take a stem cell and cause it to become endoderm, and I'll show you exactly how we did that. The next step is, once a cell becomes endoderm, have a genetic circuit that causes that endoderm cell, and only when it is ready, not before it is ready, when it is ready, cause it to take the next step from endoderm towards pancreatic progenitors. And when it's pancreatic progenitors, take the next step to endocrine, and so on and so on. So basically, it's a set of if-then statements that drive in an automated fashion this entire cellular differentiation process. So let's take step number one. Okay, just to give you an idea, the step number one took us about 10 years. Okay, I think we're getting faster now, but still, just to give you a ballpark of how long these things often take. And so this was one work that was done by Patrick Gee, former postdoc in the lab, and also with collaborators like Mo, who was a postdoc with Linda Griffith here at MIT. And so the first step was for a stem cell, if DOX, so DOX is a small molecule, then we want that cell to become endoderm. And the way we envision doing that is by producing or expressing a particular protein called GATA6. Now, when we started this, and until we demonstrated this, this was not known. But GATA6 was implicated in endoderm differentiation, but nobody knew whether if you could produce GATA6, you can drive stem cells towards endoderm in a genetically engineered fashion. So we set out to do this. So that's the kind of computer science program, small program. Here's the biological program. Okay. Now, this biological program is actually the simplest biological program that I'm going to show you today. But let me at least walk you through it. So you don't have to necessarily know all the details, but I want to mention at least the high-level details of this biological program. So at the very top, we have this genetic construct where we have a region of DNA called a promoter, so here I'm showing you PUBC, and it's a UBC promoter, which is responsible for making several proteins. One of them is the RTTA3 protein. And so it's always going to make it. And so the cell is always going to make this protein. It's a foreign protein. And when we also add this small molecule DOX to the system, it goes inside the cell. It's going to bind RTTA3, and RTTA3 activates the production of this TRET promoter, which results in expression of GATA6. This is a protein that we wanted to produce. There's also production of EGFP, which is a green fluorescent protein, so we can see what's going on, what cells are making GATA6. And there's a production of BFP, which allows us to see, it's a blue fluorescent protein, allows us to see which cells have the circuit. Okay. Now, we put this genetic circuit, so we made a piece of DNA, we put this genetic circuit in these stem cells. But what happened is that we actually put these circuits in different copy number in the different cells across the population. And as a result of that, some of the cells, when we added DOX, made a lot of GATA6, and some of them made a little GATA6. So we had really a diversity of GATA6 expression levels. Now, we didn't think this was great, but we said, let's just see what happens. We typically want to have very precise control, but here we said, this is what we have at our disposal. Let's just go ahead and see what happens. Turns out that that diversity in GATA6 expression levels was the reason for success here. So the fact that we didn't have a tight distribution, but rather we had a very wide distribution, was the key for success. And so what happened? So day 0, you see there's no green. You see the picture on the bottom, top right. There's no green across the individual cells in the population. Cells are expressing blue. There's also this red that we used here for illustrative purposes. It's not essential. And you see after 5 days of induction with the small molecule doxycycline, some cells are expressing lots of green, some cells are expressing very little green, and we see that because they're still blue and red, but they're not making green. And what happened is that all the green cells basically aggregated, and the cells that had very little green aggregated too amongst themselves. And so we can actually measure the amount of GATA6 across the population, and we see again that there's a real wide diversity of GATA6 expression levels. And it turns out that the cells that were making lots of GATA6 or medium GATA6 ended up becoming endoderm or mesoderm. Okay, so endoderm and mesoderm are two of the important germ layers, and the third one is ectoderm. So these are the germ layers from stem cells that then are responsible for making any type of tissue in your body. Okay, so we had the ability to control endoderm and mesoderm formation and also ectoderm, but let's not worry about the ectoderm for right now. And what happened is we then asked the question, once we can genetically engineer the cells to become endoderm and mesoderm, what actually happens? And so interestingly, what happens is that we basically give it the right push. And the endoderm cells now became hepatic. Okay, hepatic here means that they became, liver-like. Okay, and the mesoderm cells began to differentiate towards hemangioblasts, and then we ended up actually creating a very robust vasculature within this construct. And so we ended up getting a vascularized liver organoid, and also contained blood cells as well. And the liver organoid actually contained all the cell types that are known to exist in a fetal liver. Okay, so we're really excited about that. Now is the first, and I think actually only example of being able to start with a single population of stem cells and be able to generate all of these cell types within a liver organoid. So we then asked, so this was done and published with a 2D environment. We asked the question, what happens when we begin to grow this in a 3D environment? Does it begin to recapitulate the architecture of actual liver? Okay, and so here are some images of these liver organoids that we're able to generate, and from a single population of about 5,000 stem cells that we begin with, we can generate these very large organoids. These are vascularized liver organoids, and we're showing you day 30, and I'm showing you day 74. And by the time you get to day 74, these are really large. They're centimeters across. So this is larger by a wide margin than other organoids that are out there, and that's because we believe of this vascularization that's taking place there. We also notice that within this liver organoid, we get lots of these other important cells, such as immune cells known as Kupffer cells. These are macrophages. They're really important for understanding liver disease, as well as cholangiocytes, which are responsible for bile duct formation. So we are very excited about the architecture that's actually emerging in this liver organoid. In addition to architecture, which is beginning to resemble some aspects of a liver organoid, we also quantified liver function. And so in terms of liver function, the liver organoids that we're able to generate have albumin and urea levels that are comparable to adult liver. And this is really important in terms of beginning to understand the maturation level of these liver organoids. And this is why Big Pharma is now excited about being able to use this as a good model for your own liver and how your liver might respond to particular drugs. Okay, and we're still working on additional ways to further mature this liver. And so that brings me to this next point here, which is we're able to create this vascularized liver organoid. But as you remember, we initially set out to create pancreatic beta cells. And so what we are looking at now, besides maturation of the liver, is can we create the following capability, where rather than liver, when the cells become endoderm, rather than having them differentiate to liver, what could we do to get them to differentiate into a closely related organ, which is the pancreatic organ? Okay, so can we cause them to basically perform this action? And this would require an if-then statement. So when the cells become endoderm, the idea is, can we produce proteins that push them towards pancreas, as opposed to producing having them be pushed towards the liver? So can we create this if-then statement that will be based on the identity of the cell type? So in order to do that, we have set out to create a variety of different genetically encoded sensors that could detect the identity of the cell as it transitions from stem cell to endoderm or other cell types. And then the point is that the genetic circuit can basically activate the expression of genes that would drive endoderm to pancreatic progenitor transition. And so I'm going to show you very quickly several different kinds of sensors. One type of sensor is a protein sensor. So this was work done by a former postdoc in the lab, Velia, who now has a faculty position in Naples, and Bree, former graduate student who's now in California. And so they created this platform whereby we can genetically engineer these what are called chimeric proteins, multi-domain proteins, that can sense the presence of other proteins inside the cell, and then conditionally activate the expression of desired genes. So here I'm showing you the ability to sense the NS3 protein, which is a disease-related, a viral protein. And the notion is that when a cell gets infected, it's going to start producing NS3. And is that something we can actually sense? And so in order to do that, we basically created these two chimeras. One protein is fused to the membrane, and the other protein is going to be free floating in the cytoplasm. And the point is that when NS3 protein is present, it brings together these two chimeras, and as a result of that, one chimera cuts the other one at this TEV, cuts the TEV cleavage site, and releases this transcription factor, which goes into the nucleus of the cell and can activate expression of whatever gene we want to activate. So whenever NS3 is sensed, we can have a sense and respond mechanism. And I'm showing you here data with activation of a yellow fluorescent protein, but we've also been able to do this with killer proteins. So for example, basically you have a genetic program which says, if detect NS3, which is indicative of a viral infection, then have the cell commit suicide, right? So kill itself rather than replicate the harmful virus. Okay. Another kind of sensor is called a microRNA sensor. And the idea here is the microRNAs are important biomarkers in the cell, and they're implicated in many processes, including certain disease processes or normal development. And so we were able to create, this is work by Jeremy Gam, he was able to create a large library of microRNA sensors, 620 different microRNA sensors, and prove that they work well, and they can actually be used to sense activity inside the cell. So HEK versus HeLa cells, these are different kinds of cancer cell lines. We can actually distinguish HEK and HeLa based on these microRNAs. So what I'm going to do is I'm going to come back to these microRNA sensors when I talk about cancer application. So for now, we leave it as it is and move on to yet another type of sensor, which is a gene sensor. And so these gene sensors are mechanisms that allow us to understand and detect when gene expression is changed inside the cell. Okay? And so when the cell, for example, transitions between a stem cell and endoderm, and then mesoderm, and then hepatocytes to create the liver organoid, the cell basically transitions in terms of the expression of various genes. And so what we've done here is done some principal component analysis that looks at different genes and how they're expressed in different stem cells as they differentiate towards the liver organoid. And you see on the bottom there, on the top right, you see the stem cell population, and then you see some cells become mesoderm to the left, and some cells become hepatic to the right over time. And so what we've been able to do, and this is work by Jose, former postdoc in the lab, is ask the question, can we find genes that specifically turn on in the endoderm but not the mesoderm, and would allow us to then create this if-then statement. So if endoderm, then activate gene expression that will drive the cell towards the pancreatic lineage. And so this is data that he's been able to show. On the columns there, each column represents a specific gene, one of thousands of genes that Jose analyzed. And on the rows, we see different cells classified by specific lineages. And so these are the lineages which define the endoderm. And you can see there's lots of different lineages. There's actually, 4 different lineages, so it's not just even one single lineage. And different genes express in different ways in endoderm versus the other cell types. But importantly, there's no magic gene that just turns on only in endoderm. There's always like a little bit of expression in other cell types. So we need to be able to say if this gene is turned on, but also combine it and genes are also off, in order to get high precision over the question, is this endoderm? So we need the ability to say, to create a genetic circuit that says if gene 1 and not gene 2, okay? So let me illustrate that. Start with iPSCs. Some of them become mesoderm, some of them become endoderm. And so we know that SOC17, one of the genes, goes up as the cells transition away from iPSCs, but it goes up both in endoderm and mesoderm. So what if we wanted to only activate the gene in endoderm and not mesoderm? So we need to be able to have the statement, if SOC17 is high and this other gene, which is SEMA3E, for example, is low. So if gene 1 is high and gene 2 is low. And if so, then we want to express various genes that will drive endoderm towards pancreatic lineage. Okay, so if we have the ability to do if gene 1 and not gene 2, then in principle, we have now the ability to genetically create complex organoids and really control stepwise their developmental processes. So just for the sake of time, I'm not going to get into the details of how this is. I'll just give you a very high level. So what we were able to do, this is work by Elvira, current postdoc in the lab, is take a gene. So this would be a gene that's present naturally in the cell, and downstream of that gene, and here's what I'm showing you, downstream of that gene, we also are able to genetically engineer expression of this non-coding RNA, and we do it in such a way that it doesn't affect the expression of the natural gene, like SOC17. And what happens is that SOC17, for example, will get expressed, will be made a protein, but at the same time, we will have this non-coding RNA, which we can then use to activate additional genes, for example. And indeed, we're able to do that such that, first of all, we're able to show that when we put these genetic cassettes after a particular gene, so in this case, RPS21, it doesn't affect the natural behavior, the natural operation of this endogenous gene. And we're also able to show that the guide RNA, and this is guide RNA that binds with this CRISPR system. And so when this guide RNA is expressed, we're able to activate gene expression of a fluorescent protein. And we're able to do it in a graded fashion. So in addition to being able to activate, we can also repress. So instead of making guide RNA, we can express microRNA. And so that overall allows us to implement the biological program if gene 1 and not gene 2. And if that's the case, we can express the target gene, for example, that will push it towards pancreatic lineage. So we've been able to do this recently. We've also had success with microRNA-based sensors and actually cause a real effect in liver organoids. And that's something I don't have slides here for, but we're very excited that we are now able to have an automated second step of differentiation in these liver organoids. Okay, let me now switch gears to cancer therapy. And this is building on these kinds of logic circuits, okay? And so what's the notion here? The notion here is that in cancer treatments right now, there's been just tremendous progress using your immune system to be able to fight, tumors, right? So be able to create inflammation in the tumor microenvironment that will then cause the immune cells to be able to come in, infiltrate tumors, and be able to really wreak havoc on the tumor, right? So it's been very powerful in terms of, for example, engineering T cells to do that, releasing immune or creating inflammation by checkpoint inhibitors, and so on and so on. And so even though there's been just amazing progress there, there's still some important challenges. How can we more precisely control the inflammatory activities so that we get the good inflammatory activities, but we don't create over-inflammation so that it could become harmful? How can we deal with mutations that happen in these tumors and so on? So there's some important challenges that remain, and my argument is that synthetic biology can really help address many of these challenges by being able to control the timing, the dosage, the localization of these types of therapeutic responses. So let me give you discuss a couple of vignettes. But before I do that, what we strive for, and this is many people in the community strive towards this objective of taking control of this cancer immunity cycle. What does that mean? So it's the ability to start by creating a mechanism whereby cancer cells release the antigen. So these are molecules inside the cancer cells that can help identify these cancer cells as being aberrant or abnormal, such that dendritic cells, which end up turning into cancer antigen-presenting cells, will pick up these antigens and then migrate to the lymph nodes, whereby they will educate appropriate T cells and sometimes B cells that are able to fight cells that have these antigens. So in this case, educate T cells, the appropriate T cells, to be able to fight cells that express these antigens, and have these T cells traffic back into the tumors, infiltrate into the tumors, make contact with cells that have these antigens, and specifically kill them. Okay, so this is one of the kind of most promising directions, I would say, to being able to really address tumors. Okay, and people have been working in some very exciting ways on addressing parts of this cancer immunity cycle. So here are just a small sample of molecules that have been used in various stages along the way, and many of them have had some real impact on tumor regression and survival. And so our question is, can we employ synthetic biology to really take control over this? And we've been able, and what I'll show you is a few important steps along the way. Okay, and so in order to do that, one of our favorite tools is using RNA therapy. Okay, and so RNA therapy, as everyone knows, is now maybe the hope for rescuing the world in a timely fashion from the coronavirus, right? So this notion of using modified RNA as a mechanism to generate the spike protein in cells, which then is used to educate the immune system, so it becomes immune to future infections by SARS-CoV-2, right? So it's a very exciting technology, and we've been interested in RNA therapies for quite some time, almost about a decade. The problem, so this messenger RNA approach, it seems to be working amazing for vaccination, partly perhaps because you don't need to produce a whole lot of antigen. However, for situations such as cancer, you need to be able to make lots and lots of your therapeutic protein. The dosage really matters, and you need high dosage. So rather than using messenger RNA, what we are using is self-amplifying RNA. Okay, and this is based on an alpha virus RNA. This is a virus that has RNA that actually replicates itself. And just to note, the coronavirus is also a self-amplifying RNA virus. So it has actually very similar features to what I'm showing here. And so the notion is that this RNA goes inside the cell, is delivered inside the cell, and this could be synthetic RNA that we make. And the synthetic RNA, because it has this some alpha virus proteins, is able to go from the positive strand RNA to the negative strand RNA, and then the negative strand RNA is used as a template for making more positive strand RNA. So it's self-amplifying. So one molecule can make lots and lots of the same RNA. So that's important. And the other thing is that in addition to making the full genomic RNA, it also is making subgenomic RNA, and this is where we can put our payload. So by putting this molecule in, we can control expression of the therapeutic payload. Okay, so I'll skip this. And so and we've been able to observe really long-term expression from RNA, greater than a month in our mouse models. Okay, so in comparison to conventional payload expression, which comes in and makes some amount of RNA, or you deliver modified RNA yourself, it makes some amount of therapeutic protein. Our approach with this RNA amplification mechanism, we start off with perhaps even a single RNA molecule. The single RNA molecule replicates itself, and we can get 200,000 copies of that RNA molecule. So we are able to generate very high levels of expression of the therapeutic payload. So here's one example of using that. This is work by Ying Zhang, a postdoc in the lab, in collaboration with Daryl Irvine. And so we encapsulate this replicon, this RNA replicon inside the lipid nanoparticle. We inject that into the tumor, and then, and you see here the tumor and the tumor draining lymph node, and then the cells that got the RNA replicon begin to make the payload, which I'm indicating is cargo. But in addition to that, because this replicon with lipid nanoparticle goes in, it actually creates inflammation. Okay. Ultimately, we actually engineered this lipid nanoparticle and replicon system to kill the cell, so that the cell results in an immunogenic cell death. And this immunogenic cell death then releases tumor-associated antigens. And so from this single injection, we have this, what we're calling this all-in-one effect, where the single injection results in high-level expression of the therapeutic protein, it results in high levels of inflammation, and it results in release of tumor-associated antigen. And so the point is that we want to create a situation whereby from an initial immunosuppressive tumor microenvironment, we create a highly inflammatory tumor that has all of these immune cells that infiltrated into the tumor. Okay, so data along the way, we indeed have very high levels of payload expression from our formulation. That's what you're seeing there. We have immunogenic cell death. That's what you're seeing here. We have an interferon response. So interferon response is when the cell gets this replicon with a lipid nanoparticle, it makes these molecules, these type 1 interferon molecules, which serve as a danger signal and further amplify the inflammation in the tumor microenvironment. And these end up resulting in high levels of cytokines, these inflammatory molecules, so high levels of cytokines in the tumor microenvironment. And indeed, we can see that we're able to modify the composition of the immune cells in the tumor. So take a tumor that does not have a whole lot of active immune cells and result in high levels of these, for example, CD8 T cells or higher levels of CD4 T cells. And so we're able to really re-engineer the tumor microenvironment to have the appropriate immune cell types. And that indeed results in significant tumor regression. It results in significant survival of mice that had very large tumors. And we did this for a variety of different tumors, tumor types. So it's not just a single tumor that's affected, but including melanoma. That's one of our tumor types. And we also show that it's not just where we inject this this treatment, but also on the other tumors. So if there's a tumor on one side, tumor on the other side of a mouse, we inject it to one side, the other side, also the tumor regresses. And ultimately, we have this durable response that if the mice have been cured, if we re-inject the tumor, they reject the tumor. So we really are quite excited about this. And so this is showing that expression of payload is important. The single formulation achieved this multiple therapies, and we can get systemic immunity. I think for the sake of time, I will then quickly talk about this other vignette in cancer, and I think I may end up skipping the last vignette that I was planning to talk about, although it's up to you. So this last vignette, then we're also genetically engineering a virus to be able to infect the cells. Okay, so this virus infects cells, but in comparison to before, it doesn't right away make the therapeutic protein. Rather, this virus then asks the question, is this a tumor cell or not tumor cell? And if it is a tumor cell, then it begins to replicate such that it's replicating only in the tumor cells and not replicating in healthy cells. And that will ultimately cause these cells to die. And in addition to that, the virus secretes these inflammatory cytokines, specifically IL-12, which helps recruit the immune system so that the immune system, which is in inflammation by means of the T cells coming in, so the combination of the immune cells and the virus together work towards eradicating this tumor. And so in order to do that, we need a mechanism whereby the virus is able to distinguish tumor versus non-tumor cells. So different cells, including tumor cells, will express different genes, and they have different biomarkers. So the question is, can we think about the different biomarkers in the tumor and create this kind of logic truth table whereby a tumor that maybe has a subtype 1 will have biomarkers A and B high and C, D, and E low. And so then the question is, can we create a logic circuit using genetically encoded mechanisms that will respond and activate only weight A and B are high, and C is not high, and D is not high, and E is not high. So can we create this multi-input logic function, this 5-input AND gate, so that this 5-input AND gate will basically very precisely distinguish between tumor and non-tumor cells. And so we would only destroy the tumor cells, and we would leave healthy cells unharmed. So I'm not going to be able to talk, as expected, about the full complexity of the circuit, but let me show you some building blocks. So let's start off with being able to sense different biomarkers such as microRNA. And so we're able to be informed through bioinformatics that microRNA 141 is low and microRNA 21 is high in HeLa cancer cells. That's a particular type of cancer cell. So then the question is, can we create a genetic circuit that basically activates the killer protein when microRNA 141 is low and microRNA 21 is high, and only in that case? Okay? And so that would involve creating a genetic circuit that performs this particular logic function. Okay? And so, again, just for the sake of time, I won't get into the details of this, how this works, but this genetic circuit can detect that and activate the alpha protein when microRNA 21 is high and 141 is low. Turns out that that's not enough. So we need to actually create even more complex circuits. So this is work done by Jen G, former postdoc in the lab in collaboration with Kobe Benenson, who's now at ETH in Basel, actually. And so we created this mechanism whereby the circuit only activates when microRNA 141, 142, and 146 are low, and microRNA 21 is high, and microRNA 17 and 38 are high as well. So it's a pretty complex logic circuit that controls when the cell will actually produce the killer protein. And this indeed is able to activate gene expression in the right cell types, although it wasn't perfect. So this is work that was done, now almost a decade ago. And so while it was pretty good, it didn't have kind of the on-off capabilities that we wanted. And so what Jin, postdoc in the lab, did is he said, let me even try to improve this on-off behavior. So he took the circuit and then he added a whole bunch of regulatory motifs. So feed forward, another feed forward, another feed forward, another, and feedback motif, another feedback motif, and another feed forward motif. And so he created an actual genetic circuit that looks really messy, that what you're seeing here, but then this circuit controls the virus so that the virus only replicates under the right conditions. And indeed, it has very good on-off behavior. And again, without going into the details, it's able to have a real effect on tumors so that the virus only replicates in the tumors, does not replicate in non-tumor cells, has a real effect on mouse survival, and it has a real effect on metastasis. Okay, so with that, I will go to the summary. So I've been able to discuss programmable organoids, where this notion of engineering heterogeneity is an important design principle, and I also showed you gene sensors. I talked about the microRNA-based cell classifier that allow us to implement very complex logic functions inside the cells that control when a virus is replicating and when it expresses a therapeutic protein, such that it only does it in tumor cells with very high specificity. And unfortunately, I didn't get a chance to talk about the last vignette was how can we create circuits that operate not in hours, but in seconds. So the nice thing is that we actually have a paper coming out on this relatively soon. It just got recently accepted to Science. And so if that's something you're interested in, send me an email and I can notify you when this paper will be published, which I'm hoping will be in a matter of maybe a month and a half or two. And so with that, I will acknowledge people in the lab that worked on organoids: Patrick, Erez, Velia, and Jeremy. People that worked on cancer: Yingzhang, Fabio, Jan, and Jin. And on this last piece, the protein toggle switch, which I didn't have a chance to deepak, lots of funding resources I'm very thankful to. And I always mention my PhD advisor, Tom Knight, that about a quarter century ago got me excited and hooked onto synthetic biology, and I haven't been able to unhook myself from that since. And I want to thank you for your attention as well.
Professor Weiss, thank you so much for this. I think the right word here is enlightening presentation, not only with respect to potential applications, but also because you made us realize the background research and work involved in this field. If you allow me, first of all, we have attendees congratulating you on this excellent lecture. Mr. Mendes from the Hellenic Pasteur Institute congratulated you during your talk. If you allow me, before we start interacting with our audience, I would like to ask, I have two questions, but I'll only start with one and I will ask the second later. With respect to cancer applications, do you think there is a pattern, that there is a type of cancer that will react better, more efficiently to this approach? For example, do you think that endocrine cancers or virus-incited cancers may be better to target with this approach?
Yeah, no, it's a fantastic question. And so there is definitely differences in how easily and also how much of a difference does this approach make towards being able to cure a particular cancer. So, and this is, as you can imagine, this is a really multifaceted, situation. And so I'll mention a few of these components. Obviously, I can't be fully exhaustive, but for example, cell type specificity. Let's say like pancreatic cancer, where you really have not just a tumor, but it's a very difficult cancer to treat because you have the tumor and non-tumor cells, and you really want to just be able to target the tumor cells, but not the non-tumor cells. And so in situations where you have this very complex tissue, I think this approach is, could be highly efficacious because we can, with the logic circuits, very precisely detect what is the tumor cell versus what is a healthy cell. And then as a result of that, only destroy the tumor cells and not affect the non-tumor cells. There are other situations, many cancer situations are like that. Glioblastoma, many brain cancers are, have those kinds of features where you don't want to inject something into your brain that will kill everything, right? And that will create high inflammation. So, that's why, brain tumors have been so difficult to address, is you need something highly specific. So specificity is certainly a hallmark of tumors that I think this approach is highly efficacious, could be highly efficacious for. And we do have data. We have also started going on, with glioblastoma as well. And another thing that I want to mention is not just heterogeneity of the tumor, but mutations, right? So tumors are basically genetic mutations. And the genetic mutations in a tumor accumulate over time. They evolve. The tumor is not a static tissue. It really is an evolving issue. And so if you have an approach that works really well for a tumor in the beginning, many of the tumors will evolve, will mutate away from being able to be responsive to your therapy. And so you can have like an interim effect, but ultimately it doesn't work anymore. And so the kind of cool thing about this approach is that we have this modular plug and play. What does that mean? So we basically take a biopsy, we analyze the tumor, find out what are the biomarkers that identify this tumor from healthy cells. And actually right now, not with new technology, with our current technology, we can go from that biopsy analysis to a valid functional virus that can address that biopsy and something we could actually inject into a tumor in 5 weeks. Now, obviously, there's, this is the technical aspect. There's obviously, FDA approval, so there's a lot of complexity with respect to it, but we can create in 5 weeks a customized therapy. And imagine that your tumor mutates. We can take a new biopsy and generate a customized virus for these mutated tumor. So these are two of the other many kinds of examples.
So within this context,
you allow me,
yes, of course,
Professor Weiss. At last I have the chance to address you. Thank you so much for being with us. Great honor and a pleasure. Technical problems. We still have some technical problems, and I see people comes in and out. It seems that there is a problem with the browser over there. So sorry for that. I wasn't able to address you at the beginning. With your assistant, we said about we have 10 more minutes or more for questions, because we have some questions. 10 more minutes? Okay. Maybe I have some questions, but maybe the audience ask for...
yes, there is a question from Mr.
You may
Mendes.
raise your hand, please.
We already have a question from Mr. Mendes. Theo, if you can bring him into the stand. I don't have access.
Yeah, hold on a second. I will. Just learning how I can do that. Have you raised your hand? Please raise your hand. The first thing there is on the upper right, there is raise hand. You raise the hand, or hold on a second, from the attendee. All right, I see.
Yes, like that.
Okay.
Thank you, Mr. Mendes.
Sorry, no. Katerina, I'm gonna get back to Alexios later. Katerina, you have the...
Okay.
Now it's Alexios again. Yeah, please go on.
I think we lost Katerina.
Alexios, I gave you access as well. All right, he's coming.
Okay, can you hear me?
Yes.
Yes, we can hear you.
Okay. So many thanks for your excellent talk. I come from a physician background, so I would like to ask you, what's your feeling, or maybe?
Whoops.
Oh, I did it again.
Welcome back, Katerina.
Hi.
All right, Katerina, go on.
I'll ask my question then, and maybe...
Yeah.
Okay, so first of all, thank you so much. It was honestly an amazing talk, and congratulations for your submission as well. So I'm on the field of stem cells and differentiation into liver progenitors, and so I have more questions in terms of the first part. So a couple of you allow me. So the first question relates to your liver organoids. We do have an issue in the field of, when we are differentiating stem cells into hepatic-like cells mostly, to have kind of a sustain of fetal markers like AFP. Do you still see these kind of fetal markers being maintained in your cultures? And if that's so, are you using, let's say, further kind of transcriptional factors to push them mature further, let's say?
Yeah, a great question, and exactly right. So what we do is, when we created this initial vascularized liver organoid, it was maturing hepatocytes, but they were not fully mature. So they had certain phenotypes that resembled, like the urea and albumin levels were really good, but certain other markers were more fetal, as you mentioned. And for example, we had CYP enzyme activity that wasn't quite as high as we would like. We also measured, ADME genes. So you probably know about the ADME genes. And so those are used to quantify, another mechanism to quantify liver maturation. And we noticed that some of them were great, and some of them, like CYP3A4, for example, were not ATF5, and there's several other ones that were not quite as high as we wanted. And we were able to, and this is really recent results that we haven't, I haven't discussed that anywhere yet. but within actually literally the last few weeks, we're able to have these conditional circuits whereby once the cells become these immature hepatocytes, they activate a library of additional transcription factors that drives them toward differentiation, and only in the hepatocytes. So the mesoderm-derived cells are unaffected. So the vasculature continues to grow very nicely, whereas the hepatocytes are then induced automatically to have the next step of differentiation. And we're able to actually measure by QRT-PCR that this is really taking place, that we really have really nice expression in a conditional way, of all these transcription factors. And then we're in the process of also validating that the enzymatic activity is congruent with these increases in QRT-PCR. So our hope is that, by the summer, or hopefully maybe by the end of the spring, we'll be able to submit a paper on this and show this multi-step automated differentiation. So
that's amazing.
So if you send me an email by the end of the spring, I'll be glad to give you more information.
Sounds good. And if I may ask my second question, if that's okay. So as far as I understand, you're kind of trying to impact, let's say, lineage decisions by kind of transcriptional regulation mostly. I assume you're also kind of monitoring the media and changing that as well, the culture media. So transdifferentiation. And how fast the chemistry is that you're producing. And then also have you ever done Vivo experiments?
So I got your. I think there's a lot of echo.
Yeah.
Is there a way to people mute maybe? Okay, because there was a lot of echo, and it's still really bad. Can you hear me?
Katerina, maybe you would like to write a summary of your question? Maybe that would be better.
Maybe Alexia, if you can mute. Okay. Can you hear me? I think the echo is gone.
So I don't know how much of that you got, basically, but I was just asking whether you have tried to do transdifferentiation and any in vivo experiments as well to kind of, let's say, in animal injury models perhaps, or, induce injury even in your organ-on-chip microfluidic device.
Yeah, great question. So with let me see if I can address all the points I remember. So the first question point was with respect to the media. So we really believe. Oh no, actually even a point before that, transcription factors. So we use both transcription factors, but we also are regulating now other factors that are important, such as Wnt and BMP. And so it's important not just to regulate at the single cell level, but we now have specific genetic circuits that do multicellular regulation. We also believe in non-transcriptional control, whether it's microRNA or phosphorylation. So we do a lot of work with phosphorylation-based both sensing and control. The second point that I remember is the media. So we actually try to have no specialized media. So this is really just basal media, where we want all of the specialization, differentiation to happen by the genetic circuits. And so that's once we move from stem cell media to basal media, and we get liver organoids to develop very nicely without adding anything. Nothing special besides a little bit of sugar, right?
That's amazing.
Yeah,
that's the dream, really.
Yeah, and so by the way, we are happy to share these organoids. So we've shared them with collaborators, in the U.S. We've shared in Europe too, and so we're happy to share these GATA6 organoids. And you'll see that they're actually very easy to deal with. So our collaborators, have had an easy time growing them and getting vascularized liver organoids that last for months. And So transdifferentiation, it's a great question. We are working on transdifferentiation. I do believe that there's plasticity. I do believe that the right expression can really transform cells. And obviously we're not the the only ones working on that. A lot of people are very interested in that. I think that what we have to contribute is this conditional transdifferentiation. So, for example, if we wanted in vivo to go from alpha cells to beta cells, or from hepatocytes to cholangiocytes, we can actually, we've already designed circuits that will be able to do that. So that if you deliver something in vivo, or if you want to do it somehow in a multicellular organoid, that you can actually cause the transdifferentiation to be highly specific, and not to actually occur, not to have genes expressed in cells that you don't want to be expressed. And the last question I remember was about in vivo studies, and that's a great question. And literally, what day is today? Today is Thursday. So last Friday we began our initial in vivo experiment, and what we're actually trying to ask, and we have a collaborator at Boston University. So what we're asking is, do the single-step GATA6 liver organoids, which become vascularized, how good are they versus the multi-step liver organoids? So do we, will we see, better connection with the vasculature? Will we see better maturation? Can we help engraftment? Can we help proliferation and maintain viability? And how do these two kinds of organoids compare to one another?
And if I may ask, one on that, that's a final thing. Where do you transplant the cells, if you are allowed to share this information?
So we did this in a kidney capsule, but we're looking.
Kidney capsule, okay. Okay, thank you. Thank you so much for all your answers and a great talk.
Thank you for the questions. They're great questions.
Thank you, Katerina. Now, if we have the time, Professor, one more question from Mr. Mendes. He's the one trying to ask his question from the very beginning. Mr. Mendes, please unmute yourself.
Okay, can you hear me now?
Yeah.
Yes, we can hear you.
Okay, so even if I am an MD, I'm a physician by training, it was exciting to hear your talk. I was wondering, I have a very broad question. I was wondering what would be your assumptions or expectations about how long it will take for these findings to reach clinical prime time?
That's a great question. So let's talk about the different projects.
Mm-hmm.
So I'll go in the order that I presented them. So I think with respect to the organoids, I would say that not to the clinic, but for use for drug development. I mean, that's something that we're working with pharma now, and that's something they're already really evaluating. So that could be on the order of months, or even like a year or two, where they can really begin to understand how to incorporate that. And that doesn't go in vivo, so you can imagine it's much less complicated. I think that, so the next thing is that can we use liver organoids for in vivo therapy? And that's obviously a much longer time scale, so we need to get the in vivo results in mouse first, which I think the in vivo results in mice, I mean, that's something that we're going to get in the spring. We're getting some initial results. I would say, one thing that's nice is these are all iPSC derived liver organoids, so they can be patient specific. So we actually could, take somebody who needs a liver and, take the fibroblast, reprogram them to create iPSCs, take those iPSCs and then program them to create liver. And this would be your genetically engineered, your own liver, your own DNA. And that would reduce the complications from, any kind of engraftment complication. So for programmable liver, I don't know. I mean, it's hard to say, but I mean, certainly more than 3-4 years before we can really begin to think about it. The next one that I'll mention quickly is the self-amplifying RNA for cancer therapy. And we actually have a startup company that came out of the lab And so we're thinking, this is a matter of, not that long before we potentially can go into phase 1 clinical trials, maybe like a couple of years or so. That could be within reason. And lastly, the genetically engineered, conditionally replicating oncolytic virus. I think that will take a little bit longer. We actually, did some projections that in an ideal case, that could be like 3 years or so.
Mm-hmm.
Yeah.
I wish you every success.
Thank you.
Thank you. Professor, maybe within this framework, we should also take into consideration the changes that need to be done on a regulatory level, because competent authorities around the world, including FDA and the European Medicines Agency, would have to actually reconsider the way they're regulating these medicines, or the new medicines that will be produced with this technology, because the in vivo requirements on a clinical level will obviously be reduced, if not minimized. So
that's a really important point. And I'll give you one example of that, where I think, one specific example where I think, we've actually had some initial conversations with the FDA about this. So let's take the conditionally replicating virus. And so, it has a particular biological mechanism of action. So, FDA cares about biological mechanism of action. And the point is that if we show that it's safe and efficacious with a particular set of microRNA biomarker inputs, and we show it works really well, and we show for a particular tumor, the big question to me is if we change the microRNAs that we are sensing. So we're not changing anything about the biological mechanism of action, but we're just changing the sensors. So, can this be considered the same drug? And I've talked to the FDA about this. Not enough yet. I mean, this is still like preliminary, but it's something that they would be willing to consider, because this would not actually be the first case. So they've actually had other cases where certain nucleic acids can be grouped together as a single family of therapies that have the same biological mechanism of action, and then you don't have to apply and go through the whole phase 1, phase 2, phase 3 again to demonstrate safety and efficacy. So I think this is one specific example where the FDA is interested in a real conversation about how synthetic biology should alter the way we think about the approval process.
Yes, that is revolutionary as well, I think, yeah. So if you allow me, if I have one last question, I see no other questions from the forum. I would like your perspective, because we've been focusing on oncology, but from what I understand, there is also a great potential for cardiovascular disease. And I would like your comment. How could we use synthetic biology to, for example, to induce regeneration? I don't know, maybe for via cardiomyocyte proliferation?
That's fantastic. I think for cardiovascular, I mean, there could be real opportunities in terms of, because we've shown you that we can create vasculature.
Yeah, that's what I'm asking.
Yeah, and so, so the question is, can we, either induce your own cells, or can we, take iPSCs and then implant them so that we can somehow control, the spatial arrangements and be able to create, bypass arteries and things like that. So this is something that we're not quite ready to pursue, but it's I think it's something that we're beginning to at least have some very high level thinking about. And also with respect to the cardiovascular diseases, can we engineer mechanisms that are able to overcome and treat plaque formation? So we began to have some initial discussions and journal club readings about whether we can take these mechanisms and with certain modifications adapt them towards plaque, removal of plaques, and trying to reduce that plaque formation, for example.
Which means that. No, but go ahead.
No, that's fine.
So if that goes to the right direction, we could even be discussing for a therapeutic approach that will result in less medication needed for cardiovascular disease eventually.
Yeah.
That is amazing. Thank you so much. So, Theo, I don't see any other questions on the chat. You're muted.
Yeah, you're muted.
I have one question if we have time, Professor Weiss. Do you have All right. It's amazing. It's excellent what you are doing over there at MIT is wonderful. My question is, first of all, how let's say compared to the traditional developmental biology, going that way, this computational biology, this perspective somehow, how you speed up the process, the whole process, by what framework?
So sorry, maybe I missed the question.
My question is compared to the traditional developmental biology that takes some time in order to come up with, let's say, an outcome. If we apply the synthetic biology approach, do we speed up the process?
Yeah, I think there's opportunities for that. I would say that the circuit engineering aspect is still very labor intensive at this point. It's very difficult. Every project that we take on still takes many years, and so I think that we are getting faster and better. We want to get to the point where you mentioned computation, so we want to get to the point where we sit in front of a computer and basically type in, we want a circuit that does this, and then have liquid handling robots that are able to create these circuits for us and are able to then put these circuits into cells and actually test to see what happens. And so this industrialization of synthetic biology via computational design and automation of experiments, including the construction and observation and analysis, I think that as a framework could really alter the way we think about biological engineering.
Wonderful.
I think it really could be transformative. Yeah.
It is actually. It's quite disruptive. It's amazing to my understanding. But the thing is, in a complex biological system, how you can get, let's say, we have hundreds of different. We have protein-to-protein interactions. We have complex systems. You mentioned 3D models. Actually, we have also time. How you can manage to have, let's say, an understanding of all this process? It needs a lot of computation. It needs a lot of, let's say, it's,
no, it's very challenging.
How do you manage?
I would say, So
I can't hear you well. I'm so sorry. I can't hear myself as well. I have problems. So sorry. Please go on.
No problem. Optimal, ideal understanding, and so we work a lot with system biologists. So we do computation ourselves, but it's really important to also reach out to domain experts. So we work with systems biologists, we work with immunologists, we work with developmental biologists. So I think that connection to domain experts is absolutely critical to be successful. We develop high throughput analysis mechanisms in coordination with others. We use other people's high throughput analysis mechanisms and models. we do some machine learning as well, which is become very popular as of late. And so you really want to think about this as a convergence of many different technologies. I think, to me, the convergence of systems biology with synthetic biology is really quite amazing and absolutely essential for the success, so that we can take into account all the potential interactions, all the potential effects and side effects and so on, to really gain a better control. And I also want to mention that synthetic biology, in addition to controlling and engineering outcomes, it also has, I think, an important role in understanding, and that's via the creation of sophisticated sensory mechanisms that can tell us what's going on in a timely fashion, in a non-destructive fashion, right? So we can actually engineer circuits that we embed into cells, and these are circuits that can really tell us what's going on in real time in the cells. And also, build. There's also the learning by building notion, right? So we learn about biology by building biological systems. So I think it's really multifaceted.
Thank you so much. Thank you so much, first of all, for having allowed us to abuse your time.
Oh, yeah.
I think that we need to close this session now. We really want to thank you. It has been an honor and has been such a great pleasure having this vivid discussion with you. With respect to our audience, I would like to remind you that next week we will have a second session on synthetic biology from Professor John Love from the Exeter University. Thank you so much, everyone, for joining us tonight. Thank you, Professor. It has been such a great pleasure. Thank you so much.
Thank you for
Good evening, everybody.
Thank you so much. Cheers. Bye-bye.